Welcome to our webinar, everyone. My name's Ron Myers. I'm the Director of Editorial here at JoVE, and today we have Wilber Sabiiti. So he published with us in 2020 on a tuberculosis molecular bacterial load assay. And today he's going to be talking about some recent updates to the technique and, some variations. We will be recording the webinar, and we'll send the recording to you afterwards. Also, feel free to add any questions you may have during the webinar in the Q&A, and we will address those at the end. Okay, take it away, Dr. Sabiiti.
Thank you very much, Ronald, for your kind introduction, and thank you to the audience from wherever you are in the world. I am grateful for your time with me today. So I'm Dr. Wilber Sabiiti, Principal Research Fellow in Medicine, or Associate Professor at the University of St. Andrews School of Medicine. And I wanted to start with this picture here. This young man on my right is actually Professor Stephen Gillespie, who's at the same time now is giving a lecture on biomarkers for the Unite for TB Consortium, which is a global consortium for fighting tuberculosis. And he is one of the first kind of brains behind the tuberculosis molecular bacterial load assay, and has done research on tuberculosis for over 30 years. And behind myself, I was demonstrating how to conduct MBLA to a number of learners from different parts of the world, 16 countries represented in St. Andrews. And the lady behind me went on to establish the MBLA in Ho Chi Minh City, Vietnam, and published a number of papers. So my presentation will cover the tuberculosis pandemic. Why do we need to monitor treatment? The current and future diagnostic and treatment monitoring methods. The tuberculosis molecular bacterial load assay, so look at its methodology, the applications and outputs so far, and the take-home message. So tuberculosis is caused by Mycobacterium tuberculosis bacteria. On the right here, there is an image of tuberculosis bacteria. This is an electron microscopy image taken after heating the tuberculosis bacteria at 95 degrees for 20 minutes. So you can see that they're hard not to crack. So even after 95 degrees of heating, they still look quite in good shape. So inside the cell are intracellular lipid bodies, which are very important for the bacteria in terms of how it prevents itself from killing by antibiotics. And then it's mycolic cell wall, which is also an important resistant mechanism for the pathogen. So the disease, tuberculosis disease, has been here for over 2,000 years. And the WHO 22 report, Global Tuberculosis Report, said that over 10 million people fall ill because of this disease, and over 1 million die per year. So that is what we call the active disease, the one that comes with symptoms. But there's another form of the tuberculosis infection which does not show symptoms. It could be subclinical, in which case the symptoms are not seen, and the person maybe has internal pathology, or it can be what we call latent, whereby the infection is there, but there are no symptoms. And this constitutes a majority of TB infection. a quarter of the world population have silent TB infection. So the rate of, the WHO has a target to eliminate tuberculosis by 2035. But the rate at which we are eliminating tuberculosis is quite slow, very miserable. And if some modeling shows that if we continue at that rate, we may need over 100 years to eliminate tuberculosis. So, and there's now an increase of drug-resistant tuberculosis, which is now adding complications into the response to eliminate tuberculosis. So the World Health Organization called for effective diagnostics to increase case detection and effectiveness of treatment. So why should we monitor tuberculosis treatment? One, it will help us detect the patients who are likely to fail treatment quite early, and then give them appropriate management or appropriate prescription, or make sure that they are closely monitored or managed to make sure to avoid losing their lives. Two, it ascertains that the treatment is working effectively for the patient, because in many settings, treatment goes on and the patient is not aware whether it's working for them. Even the doctor is not aware, but they say, okay, you have 6 months of treatment, just go on and take the drugs. But we are not, but if we have good treatment monitoring methods, then we know whether the treatment is working for the patient. Again, prolonged stay on inappropriate therapy is a recipe for emergence of drug resistance. So the more the person stays on the medicine which are not working, then that's a recipe for emergence of drug resistance. With good treatment monitoring methods, we can prevent premature discharge of patients and subsequently reduce retreatment. Because as we know today, the majority of drug-resistant tuberculosis cases come from people who are returning for treatment, or the ones we call previously treated patients, or the retreatment patients. So, and that could be that maybe they were discharged before the disease was cleared or well managed. So with good treatment methods, monitoring methods, we can be able to discharge patients when they have cleared the disease fully. And then good treatment monitoring methods can generate data, which is important for post-registration surveillance of new medicines. So there are a number of diagnostics that are available for tuberculosis, and some of them double as diagnosis and also treatment monitoring. Smear microscopy is one which is used for diagnosis at the same time for treatment monitoring. Culture, both solid and liquid, can be used for diagnosis at the same time for treatment monitoring. Then we have DNA-based molecular methods, which includes TB-LAMP, TrueNut, line probe assay, expert MTB-RIF, or expert MTB-RIF Ultra, which is the newest version of this technology. So those are the DNA-based molecular methods. Then we have the RNA-based molecular methods, and there are two types of this. The pathogen-specific, like the tuberculosis molecular bacterial load assay, and we will be discussing more details about it today. And then the host TB infection-specific methods. For example, there's the expert host cartridge, which is now under evaluation. And there are some new assays in this field that are coming up. There are other diagnostics that are in the pipeline to diagnose tuberculosis. So smear microscopy is I think, the oldest diagnostic for tuberculosis, invented around 1885. It's cheap, affordable, and is used in many resource-limited settings. Time to results is shorter than culture, but the challenge is that it is less sensitive because at least we need around 10,000 colony-forming units per mil in order to diagnose tuberculosis confidently using smear microscopy. Analysis is subjective because sometimes if somebody is a new technician, they may confuse artifacts as TB bacilli. So the more experience you have, the more accurate you become. And it does not distinguish between dead from live bacteria in a clinical sample. So for example, if treatment has started, the microscope will not distinguish whether the bacteria are still alive or whether they are dead. So solid culture is also the oldest form of culture in the diagnosis of tuberculosis. It uses the principle of multiplication. So starting with a few cells at the beginning, inoculated on the plate with a good medium, which I've called food. And then the bacteria grow, they multiply, and then they form colonies, which can be visualized on a plate. As you can see here, the LOGS lands with colonies of TB, and then these other blood agar, the plates with colonies. It was first described around 1905. It is used for detection and also for quantification of bacterial burden. I think before the invention of TB-MBLA, solid culture was the only way of quantifying tuberculosis bacillary load by counting colonies on the plate. But as we know, tuberculosis grows very slowly, and so it will take weeks before colonies can appear for counting. It's less sensitive because of the slow-growing TB bacteria, and it's now established that not all TB bacteria grow on solid culture. And contamination can also be an issue. Then we have the liquid culture, the Mycobacterium growth indicator tube or midget culture system. This is a fully automated non-radiometric system, which was first used in the 1990s. It uses the principle of oxygen consumption by growing bacteria, which triggers a fluorescence signal that is detected by the machine. So as the bacteria grow in the medium, they consume oxygen, and that consumption triggers a fluorescence signal that is detected by the midget machine. It's more sensitive than solid culture, and the time to positivity is inversely correlated to bacterial load, as we shall see shortly in the ensuing slides. So the higher the bacterial load, the shorter the time to result. So that means the result is highly dependent on the bacterial load. The time to result, or the turnaround time, is highly dependent on the bacterial load of the sample. So if two patients report today, one has high bacterial load and the other one has a low bacterial load, the one with high bacterial load will get their results faster than the one with low bacterial load. And we also know that a proportion of TB bacteria do not grow in the liquid culture. They will need a supplement, a resuscitation promoting factor, for them to be able to grow. So that means the quantification is not comprehensive because some of the bacteria are not growing. The other challenge for the liquid culture system is it is very prone to contamination, and that means there's need to use sodium hydroxide to decontaminate the sample, but that again kills some of the TB bacteria and then compromises the sensitivity of the midget in terms of diagnosing and monitoring treatment response. The molecular methods, in this I would single out the gene expert technology, which is now the widely used technique. So for some countries, already in many parts of Africa and parts of Asia, it's now the first line diagnostic for tuberculosis. It's more sensitive and it's rapid. The results can be obtained in 2 hours. It's not affected by contamination because it's a PCR-based system, uses specific primers. It detects also rifampicin resistance, which has been an advantage for the system because now there's more rifampicin resistance that is detected across the world. It's a modular system, and that limits the number of samples that can be tested in one go. And so, for example, if a clinic has a 4-modular machine, and then they get 16 patients, it means that they need over around 8 hours in order to process all the samples, because each 2 hours they can only process 4 samples. It detects DNA, and therefore it cannot distinguish dead from live bacilli, because DNA is a very stable molecule. It can survive long after cell death, and so it's not a good marker for live cells. So it's not suitable. The gene expert technology is not suitable for treatment monitoring. So this brings us to the tuberculosis molecular bacterial load assay technology. So it's based on the ribosomal RNA of prokaryotic bacteria, which is a structural RNA made of different units. And we use one of the units, the 16S unit, for detection of Mycobacterium tuberculosis and its quantification. So we know that RNA is only present in live bacteria, or it's degraded soon after the cells have died. So that means we can distinguish between dead from live bacteria. Why do we need live bacteria? It's because if someone is on treatment, we want to know that the medicine has killed this number of bacteria, and only these bacteria are left, or they are still alive. So that means the treatment should continue until all the bacteria have been killed by the medicine. So that's why we specifically want to measure live bacteria. The ribosomes are so many, around 700 ribosomes per centimeter cubed in the cytoplasm. And that means that we have higher sensitivity to detect even very low bacterial load samples. So it can detect both actively growing cells and dormant bacilli. TB has this character whereby there is this proportion of the bacilli that are actively growing, and there's another proportion that is not growing. It's just like a backup. And normally the one that is not growing is the one that becomes persistent and sometimes turns into drug-resistant forms. So, but with MBLA, you are able to quantify all of them because we can extract all the RNA from the both forms of TB. So time to result is 4 hours from when the sample has arrived in the lab. And the turnaround time does not depend on the bacterial load of the sample. If 2 patients come now, the one with a low and high bacterial load, they will all get their bacterial load quantified at the same time. So the principle just diagram in a kind of a pictorial way is so before treatment, we assume that there are many cells in the sample, many TB bacteria in the sample, and inside the TB bacteria there are ribosomes, which are the ones that our assay targets, and then when treatment begins, then these cells are reduced in number, so we are left with fewer cells, and that means less amount of ribosomal RNA that we can quantify. So when compared to the liquid culture, immediate liquid culture, what we see is that there is an inverse correlation between the TB-MBLA measured bacterial load and the immediate culture time to positivity. So the red bars is the immediate, is the TB-MBLA bacterial load, and then the black bars and curve is the time to culture positivity. So when the time to culture positivity is increasing, then the bacterial load is decreasing. So the lower the bacterial load, the higher the time to culture, or the longer the time to culture positivity. So there is an inverse relationship between the two, and that shows that actually they are all measuring live organisms. The difference is that TB-MBLA will deliver results much faster, and it's not affected by any contamination, and means that the doctor can get results in good time to make decisions and manage the patient efficiently. So this is kind of a summary of how the technology works. It detects and quantifies viable Mycobacterium tuberculosis bacilli. The assay consists of 3 main steps. The first step is isolation of RNA from Mycobacterium tuberculosis cells. Second is quantification of this RNA using the reverse transcriptase quantitative PCR. And then the third step is conversion of the PCR output or the cycle threshold values into bacterial load using a standard curve. And ECFU means estimated colony forming units per mil. So how do we isolate RNA? So this, we start with a sample from the whatever vessel has been brought in. And at this stage, an extraction control is spiked into the sample. Then centrifugation is performed to harvest or sediment the cells. And then this is followed by lysis. We conduct mechanical lysis using homogenizers. These are on the side, on the right side are the 2 homogenizers, the Predictive 24 or the MP Biomedical's fast prep machine. They are the most common that we use to mechanically homogenize the TB bacilli and lyse them. So once they are lysed, the next steps is to purify the extract by removing the lipids, proteins, and other cell debris. And this happens by chemical purification methods. So there are 2 types of kits that are commonly used: the phenylchloroform kit from MP Biomedicals, or the column-based kits from Qiagen or from Thermo Fisher in Vitrogen. Then so this step leads us to nucleic acids. So here we have both RNA and DNA still in this extract. So we conduct a DNase treatment whereby DNA is digested using DNase enzyme and leaving us with the RNA extract that we can take into the PCR right away or store for short or long term. Short term, you can keep it in -20 or just in the fridge, or long term, you can keep the RNA extract at -80 degrees. So here is just for us to appreciate the different RNA isolation techniques that are available out there. So for now, we've been mainly using the manual extraction kits that are available commercially. These fast prep, fast RNA per kit, the QIAGEN RNase, the pure link from Invitrogen, and a number of other total RNA isolation kits can also do the job. But you need to make sure that you have the ELISing matrix. The beads are very important to break open the bacilli. Then there's the semi-automated system. So this is a Thermo Fisher extraction robot. We are now evaluating this to find out if it's effective in extracting RNA, and this can be helpful for high throughput labs that have many samples to run. So instead of doing manual extraction, just put in the sample and you just come for your RNA after. The future is to build a fully automated system whereby RNA extraction and RT-qPCR all happens in one compartment, in one system, without having to put your hands on the sample. But the advantages of both systems. So the open system, whereby it can apply, you can extract anywhere, like open manual extraction, is good in a way that you can extract from all sample types. So the inhibitors that normally inhibit are not a problem in a manual extraction system. Automated or fully automated systems, sometimes the chemicals used in the extraction can inhibit the PCR, and that limits the number of the sample types that you can use. So there are advantages to both systems, and we plan to keep both. Those who want to use the manual or those who want to use automated can use. So the PCR step, which is the next step after RNA extraction, involves four types of samples that are being run. Some of them are controls. So we have the MTB and the extraction control standards. These ones are used to construct the standard curve that is needed for conversion of the quantification cycles to bacterial load. Then a positive control, which is also supplied with the kit. This is to control for the PCR, to make sure that if you've got a negative result, it's a negative because your positive control has worked effectively. Then the negative control, which is normally the water that you have used to prepare your master mix or your primers, that is important to control for the cross-contamination in the lab. And then the patient samples that you want to determine the bacterial load. We also do what we call the reverse transcriptase negative reaction. This is important for ascertaining if the DNA removal process was effective. Because in this reaction, we do not add the reverse transcriptase. So if there is any signal that is amplified in this, it will be a signal that is coming from genomic DNA. And that means that our process of removing DNA was not effective. So there's several controls to ensure that the result we get is trustable and it is from RNA, which is a marker of viable bacteria. So the PCR can be performed on various platforms. It's the Rotagen machine supplied by QIAGEN. There is the VIA7 and Quant Studio from Applied Biosystems and Thermo Fisher. And then there is this system from Roche, and other PCR system, real-time PCR systems can be used to run the TB-MBLA PCR. The master mix, we have now made it easy. This is a new update because the last time we published with JoVE, we were using this kind of a manual master mix whereby the user would have to make up the master mix themselves and then run the PCR. So we have now developed a ready-to-go master mix, so with everything in it. So what the user does is only to distribute the master mix into the reaction tubes, 16 microliters per reaction, and then add the 4 microliters of the RNA extract. That makes a total of 20 microliters per reaction. And then they are ready to go for the PCR. So what we normally advise the users of our kit is that they will receive the RT positive, the reverse transcriptase positive mix, and the RT enzyme that is in water, and that they're required to do is to mix the two and then make a complete mix for reverse transcriptase positive reaction. And then the reverse transcriptase negative mix is supplied also separately. So we advise that once you receive the kit, make small aliquots of 200 microliters and store them at -20 for further use. This will prevent frequent thawing and also prevent cross-contamination. At least if one vial is contaminated, then you have a chance to throw it away and then use the vials that have not been opened. So on the day of the PCR, you take out the quantity that you need, and then you make your master mix and run the PCR. So these are the PCR conditions. The 30 minutes at 50 degrees for the reverse transcription to take place, and then 15 minutes at 95 for DNA denaturation, then primer annealing 45 seconds, then elongation 60 seconds, and this happens times 40 cycles. We've also worked at means of shortening the PCR reaction from 2.5 hours, and now we have a protocol that can achieve the PCR reaction within 1 hour. That reduces the time taken for the reaction from 2.5 to just 1 hour. So once the PCR reaction is done, this is what you expect to see. These amplification curves, beautiful sigmoid shaped curves that show that the amplification has taken place. And then this with the Rotagen machine, it has a software that will correct the curves and the slopes of the curves so that they are good to be analyzed, because the threshold line should go through the middle of the exponential part of the curve, so that you have a balanced sensitivity and specificity. If the threshold line is lower, you're likely to be to lose specificity by picking the background amplification. And if the threshold curve line is too high, it's going to compromise sensitivity by losing some of those low bacterial load samples. So very important. So we've optimized this, and it's at 0.01 threshold. So we have different thresholds for different PCR platforms. So applied biosystems platforms, for them the slope is automatically corrected, so you don't need to do anything about it. You just need to set the threshold for those platforms. So for users who have such platforms, they normally write to us, and then we tell them the threshold, and we give them SOP for their systems. And then this is the standard curve. So on the x-axis is the concentration from our standards, and then on the y-axis is the CT values or the quantification cycles, and there is an inverse relationship. So the higher the lower the bacterial load, the longer it takes for the sample to amplify, and vice versa for higher bacterial load samples. So this is a sum up of the whole process. So we begin from the sputum collection. If your sample of use is sputum, and then RNA extraction, followed by the qPCR, and then finally the analysis of the PCR output and getting the bacterial load results that you can now send either to, if it is a clinical trial, put into the database, if it is for the clinic. I want to note that TB-MBLA is still a research use only kit, so we are not yet using it for routine clinical work, but that's the next step. And I think plans are already underway to get TB-MBLA as one of the routine kits. So this is the interpretation of the qPCR results. So if the MTB channel is positive and the extraction control channel is positive, then the result is a valid positive. If the MTB channel is positive and the extraction control channel is negative, then the result is okay, but we cannot use it for quantification because we are not sure whether the extraction was efficient since we have not retrieved the extraction control. If the tuberculosis channel is negative, the extraction control is positive, then that is a valid negative result for MTB. If the MTB channel is negative and the extraction control channel is negative, then that result is invalid. So the sample needs to be repeated. So we've done some work to, now this brings me to the applications and the utility of TB-MBLA. The first work we did was to find out whether the TB-MBLA is reproducible in different laboratory settings. And this was work done in different laboratory sites in Africa. And the laboratories were supplied with standard panels of TB bacteria, the one with high concentration and the second one with a low concentration. And they all got consistent results from each other, indicating that the assay is reproducible in different laboratory settings. We've recently done some work comparing the TB-MBLA and other methods. We compared it with the GeneXpert, which is the black curve here, and then the green curve is the smear microscopy, and then blue curve is immediate culture. And what we found is that TB-MBLA is consistent with culture in terms of monitoring treatment response, and the response is biphasic in nature, very rapid clearance in the early days, and then slow clearance as in late after 2 months of treatment. But what we found is that while other methods responded quite rapidly, the GeneXpert responded quite slowly. And this, further investigation, we found that at the end of the day, at the end of 6 months, 33% of the patients were still positive with Expert Ultra. And we followed them further up to 9 months to see what happens. And at 9 months, all of them had cleared their TB and had no clinical signs at all. And the post-treatment follow-up, there was no more medicine given to the patients. So that means that there's a lot of accumulated dead DNA in the during treatment, which can be picked up by Expert MTB Ultra, that again makes treatment monitoring difficult. So we found that it takes 14 weeks to clear ribosomal RNA compared to 52 weeks for DNA in the same patient. So DNA, you'd need at least a whole year before you know if the patient has responded to treatment. So the other importance of TB-MBLA is to be able to show us the patients who are responding and clearing bacteria quite early. Because what we found in this study was that as early as 2 weeks in treatment, there were patients who converted to negative and they never reverted back to positive for the whole 6 months of treatment. And 19 of these were detected by MBLA, compared to 15 by immediate, and then 13 by smear microscopy, and only 7 by expert MTB reef. So that means that some patients clear early. So should we keep them on treatment? And we checked whether maybe they started with a very low bacterial load. Actually, the bacterial load was not that very low. They were average around 40,000 colony-forming units per meal. So this means that some patients have ability to clear very fast. And this is an advantage in case we begin to develop personalized treatment for tuberculosis. Then we can know patients who have cleared, and then we don't need to follow them up or to treat them for the whole 6 months. We've also done some work using drug-resistant tuberculosis. This is a cohort of patients recruited in Nigeria, Ethiopia, and South Africa. And what we show here is that if you compare rifampicin monoresistance, RRTB, to multi-drug-resistant tuberculosis, MDR-TB means rifampicin resistance plus isoniazid resistance, or any other forms. And we see that rifampicin monoresistance is resistant bacteria cleared faster than the MDR-TB bacteria. And this on the side are the regression lines to measure the slope at the rate at which the bacteria were cleared. Then we compared this drug-resistant TB cohort with a drug-susceptible TB cohort from Uganda to see how they compare in terms of treatment response. And what we see here is that the drug-susceptible TB starts with a very high bacterial load, higher than the drug-resistant TB, but by week 2 of treatment, it has overtaken the drug-resistant TB in terms of response to treatment. So, meaning that the drug-susceptible bacteria are cleared faster than the drug-resistant TB bacteria. So, analysis of the hazard ratio of how the risk, we found that if a combination of high bacterial load and drug-sensitive tuberculosis has a risk of 2.7 for not converting to negative by month 2 of treatment, compared to 4.9 for drug-resistant tuberculosis. So a combination of high bacterial load at the baseline and drug-resistant tuberculosis will increase the risk of not converting to negative during treatment. And this is the advantage of TB-MBLA in a way that it can provide us the means to detect the patients who are at risk of not succeeding in treatment, and then they are given appropriate management. So there are other applications of the TB-MBLA. We've already shown that it's better than culture in terms of predicting patients who are at the risk of relapse of treatment, of disease relapse. We've also found that TB-MBLA is quite good in terms of distinguishing drug regimens that are more efficacious in clearing TB bacteria. We've also shown that this can reduce the cost and the length of clinical trials by offering rapid and objective data. We've also found that MBLA can be applied to other sample types, for example, stool, where we've shown over 80% sensitivity and specificity in stool samples. Colleagues in Vietnam have applied it to cerebrospinal fluid, and they have shown high sensitivity. And it has also opened research possibilities for preclinical drug development in animal models. For example, colleagues in Barcelona, Spain, are now using the TB-MBLA to monitor for their Drosophila model of drug resistant of TB drug development. So, as I said before, MBLA is still a research use kit, and it's being applied widely in clinical trials, a number of clinical trials. The ones that are now ongoing is the Triad Consortium, then the TIME trial, the Unite for TB trial, the PAN-TB, which is in the US, Latin America, and some parts of Asia, and in the OptiRemox, that is in Africa. Unite for TB is all over Europe, parts of Latin America, USA, and also Africa. We also do training groups. We go to different places and train people on how to implement TB-MBLA, and we've trained over 500 people in how to apply TB-MBLA. So where can you get a TB-MBLA kit if you need it? So University of St. Andrews can provide the kits if you need them. There is you can write us an email or go to the website, the Vital Bacteria website, and you can order for the kit. The University of St. Andrews has licensed a company in Edinburgh, UK, to commercialize the kits. So this company is also manufacturing design-locked kits for TB-MBLA. So if you say you have a clinical trial, a large study, you can get in touch, and we can put you in touch with LifeArc, and they can see if they can provide kits for you to run your study. The take home is the TB-MBLA offers a more effective alternative for measuring treatment response. TB-MBLA increases the possibility of implementing shorter treatment regimens for TB. For example, the World Health Organization recently approved the 4-month treatment regimen for drug-susceptible TB. So TB-MBLA will come handy in terms of ensuring that this shorter regimen is indeed helping patients to clear the disease and they are not relapsing. TB-MBLA increases the possibility of diagnosing TB in different sample types, which is, again, crucial for pediatric TB patients. For example, diagnosis of TB in stool would be very important and easy for pediatric patients. Thank you very much. I want to end by acknowledging all the team that have been involved in developing, generating this data, and the funders that have funded our TB-MBLA research from UK, Europe, and other parts of the world. Thank you very much.
Okay, thank you for the excellent presentation. So if anyone has any questions, feel free to add those in the Q&A section and we can address them. Dr. Sabiiti, it looks like there's one question there already.
Okay. Is that in the Q&A?
Yes, it's in the Q&A.
Okay. The question is, any insights how digital health can be leveraged in TB control and care? Yes, I think digital health is important, and it should be the way forward in terms of collecting and managing data. Because, for example, MBLA is generating this good quantitative data of how patients are responding to treatment. So if this data is gathered from different facilities and well managed in databases, then analysis and modeling can be done, in which case biomarkers can be developed to say, okay, if somebody has a bacterial load of this amount, then they are at risk of relapse or they are at risk of failing treatment. So I agree, digital health is critical in terms of getting that critical mass of data that can inform modeling and also the use of artificial intelligence. Then the next question is, how many samples can be run at a time? So it depends on the type of machine you're using. So a number of real-time PCR sample machines, they can run from 36 up to 384 samples. So if you're using a Rotagen machine, you can run up to 100 samples at a go. If you are doing applied by a system like Considio, you can do 384 samples at a go. The other question is, can MBLA detect drug resistance in MTB? No, we do not detect drug resistance directly, but we detect if the patient is not responding. If the patient is not responding to treatment, that means the bacterial load is not decreasing or it is increasing, then that triggers the next assessment to find out, is it drug resistance? Is it because of poor adherence to treat to the medication? So it gives a chance the doctor to ask these questions quite early and give the patient the right form of treatment. I would want you to go over the interpretation of positive and negative in the table, especially the invalid. Okay, in the interest of time, I may not go to that slide again, but what the invalid result is if the TB channel, the MTB channel is negative and the extraction control channel is negative, then it means that we are not sure whether the negative in MTB is because the extraction process was not effective, because the extraction control has not been retrieved from the sample. So that makes the result invalid. So to make sure that the result is valid, the extraction control channel must present a positive result. The next question is, thank you for a great talk. I work as a TB clinician and have two questions. As the MBLA measures messenger RNA, is there a problem with stability of RNA? To handling of the sample, how many samples can be analyzed at the same time within 4 hours report time? Okay, good. The first question is, MBLA measures ribosomal RNA, not messenger RNA. So messenger RNA, yes, is quite unstable, but ribosomal RNA is a bit stable, and it's not as sensitive as messenger RNA, so that we are able to retrieve it and run the PCR without panicking. How many samples? As I said before, it depends on the type of machine you're using. Within 4 hours, I can say for very skilled technicians, they can run maybe 10 samples within 4 hours, including the extraction. And if they have the automated system, then they can do even 100 samples in that time. The next question, have you tried to use samples different than sputum with TB-MBLA method? Nice talk and thank you. So yes, we have tried MBLA on different sample types. We have done it on stool, especially from adult stool. We want now to do it from the pediatric stool. We have also done it on cerebrospinal fluid. Some people have done it on lung tissues from some of the animal models, mice and guinea pigs. And then we have colleagues in Barcelona who are running it on the fruit fly to quantify bacterial load from the fruit flies.
All right. Well, I think that is the end of the questions. So we will be sending out an email with a recording of the webinar. So you have that for your records. And if you have any additional questions at that point, you can feel free to respond to the email, and we can do our best to help you with those questions. Yeah, so thank you to all the attendees. It's great attendance at the webinar today, and thank you for the excellent presentation. And yeah, we'll send out the email later in the week, and we'll take it from there. Thank you, everyone. Greatly appreciated.
Thank you very much. Thank you.